Great. Well, good afternoon. I'm really excited to be here. WindMIL is a very new company that's working on technology that's been under development for quite some time at Johns Hopkins. But the company was formed a little over a year ago, and this is actually one of our first public presentations. So I'm very happy to sort of be out here talking about WindMIL, and I hope you'll be hearing a lot about this technology in the coming months to years. So WindMIL was created on the work of Kimberly Noonan and Yvonne Borrello, focusing on the bone marrow. The bone marrow is a very special compartment in the body that is the natural reservoir for memory T cells. So our belief and the evidence that we have and others have shown that in cell therapy, the cell source makes a big difference. Most people are working with, for oncology indications, are working with cells from the periphery, either the peripheral blood, which is, we all know is the CAR-T and ETCR approach, or the TIL approach from the tumor site. The bone marrow actually has some very unique qualities. In a normal immune response, upon the initial encounter of a T cell with its cognate antigen, a population of memory cells is formed. Those memory cells migrate to reservoirs, including the bone marrow, where they persist for very long periods of time to be reinvigorated upon reinfection with that same insult. Doctors Borrello and Noonan postulated that perhaps if a T cell has ever seen a tumor antigen, there would be a memory population that would be formed that could be found in the bone marrow. And that's indeed what they proved. And these cells have some very unique characteristics when you look at the cells from cancer patients. Number one, they have built-in tumor specificity. So their native TCRs recognize tumor antigens from the cancer type that the patient has. Number two, they have increased cytotoxic potential, both in cancer systems as well as in independent research from other labs showing in model systems such as viral antigens, et cetera. Memory cells from the bone marrow are better cytotoxic generators of cytotoxic killers than memory cells from the periphery. And number three, they persist for very long periods of time. There was a publication from Dr. Noonan and Borrello at last year's ASH showing they could detect these cells as long as they measured, up to 9 years after reinfusion in their phase 1 trial. So WindMIL was created to bring these cells forward as a treatment for cancer and license the process, both patent and trade secret, as well as associated IP, that will allow us to activate and expand these cells, which are called marrow-infiltrating lymphocytes, or MILs. So when you think about WindMIL, it's really a company that have MILs at the center, and we can take these in different directions. On the left, we can use the cells after reactivation and expansion in unmodified form, so no genetic modification requires. We take advantage of their inherent tumor specificity. They are very low in toxicity. They've been in over 100 patients, and they have very low-cost manufacturing in relative to other cell therapies because of the lack of genetic modification. So I'll describe to you the at least 2 programs we have with unmodified MILs. In addition, we think that MILs are a better cell source for a variety of genetic modifications. And we bucket those into modifications that are already out there, for instance, the CAR-T modifications, as well as our own proprietary modifications. So this leads to a very broad pipeline. Before the company was even formed, the folks at Hopkins had begun a randomized, multi-center phase 2 clinical trial in high-risk myeloma. We are preparing to start a phase 1/2 trial next year in lung cancer. We're examining 2 other solid tumor types preclinically, and then we're working preclinically on the genetically modified side. So that's an overview of WindMIL. Given the short time, I just want to give you a flavor of some of the data. This is data that led the Hopkins group into phase 1 trials. So this is preclinical data. And in both cases, on the left, which is in vitro, and on the right, which is in vivo, in animals, in all cases, the effector cells used, either PBLs or MILs, are derived from myeloma cancer patients. So on the left, what you have is human either PBLs or matched MILs, either activated or unactivated, and we test for proliferation in response to autologous tumor, CD138 cells, or a negative control, an autologous cell marked by CD33. And it's very clear to see that the only cells that respond are the activated MILs, suggesting that in the bone marrow of these cancer patients, they have memory T cells that can recognize tumor antigens. On the right is a NOD-SCID animal model where a human myeloma cell line was introduced, and the mice were treated with either activated PBLs or activated MILs. What you can see is that only the activated MILs can control the tumor. An interesting note that's not shown on this slide is that upon sacrifice of the animals in the PBL group, there was no evidence of human CD3 cells. So the cells that were introduced were completely gone. In contrast, at the end of the experiment, when the cells in the animals in the MILs group were sacrificed, there was evidence of persistent CD3 cells. Again, speaking to the fact that these cells are naturally designed to persist for long periods of time. On the strength of that data, there was a phase 1 clinical trial that's been published. A couple of these were patients that were getting ready for transplant but were not able to reach a CR before transplant. Reaching a CR before transplant is the best predictor of CR after transplant. A couple notable things about this trial. Number one, there was a 100% success rate in expanding the MILs, so this is a very robust process. Number two is a very benign safety profile that was witnessed. No cytokine release syndrome, some mild GVH. Number three, there were a significant number of responses, but I'll quickly draw you to number four because this was a single-arm trial. The thing that was so compelling is that the clinical response was correlated directly with the activity of the MILs that were put into the patients. So MILs that were highest in memory cell content, highest in interferon production, highest in a variety of other activation markers were the patients that experienced a complete response. So on the basis of this, a phase 2 clinical trial was designed in multiple myeloma, and Dr. Borello and Dr. Noonan were very interested in using this early in myeloma when the immune system has not been so deteriorated by the tumor. And so what they decided to do to run a reasonably length trial was to go into a high-risk population. These are patients that, despite the advent of many great therapies in multiple myeloma, still have a relatively short survival, in the progression-free survival on the order of 1 to 2 years. So 110 patients were enrolled across, or planned to be enrolled. We've enrolled about 100. Across 4 different centers. All patients are defined as high risk by cytogenetic abnormalities. All patients are going for transplant. Everyone gets induction, everyone gets their MILs harvested, everyone gets a transplant, and then one of the 2 groups gets MILs, and everyone goes on Revlimid until progression. As I said, the trial is 90% enrolled. And we're very excited to have a brand new company with an ongoing phase 2B trial, which is something we don't see that often. So let me shift a little bit to just a quick word on solid tumors. So obviously myeloma is a tumor that infiltrates the bone marrow. But if you think back to my original description of how the bone marrow is involved in the generation and the maintenance of memory, the immunologic memory, the theory is that you don't have to have bone marrow, tumor in the bone marrow to find these cells, because they are a group of cells that once antigen is seen would migrate into the bone marrow and persist there for long periods of time. So indeed, what we did is we tested bone marrow from lung cancer patients and looked at whether or not, number one, we could grow MILs because their bone marrow microenvironment is different, and so that process has been worked out. And number two, we looked at whether or not these cells could recognize common lung cancer antigens represented by lung cancer cell line lysates loaded into autologous antigen-presenting cells. And in all of the patients we've tested so far, that is indeed what we see. Interestingly, the negative control in this experiment, in addition to just straight media, the negative control is myeloma. So I showed you that myeloma patients have T cells that recognize myeloma. We would hope, if our theory is correct, that lung cancer patients do not recognize myeloma, and indeed they do not. So all 4 patients we've tested have tumor-specific T cells. Interestingly, 3 of these 4 patients had non-metastatic disease. So we're going to be planning a lung cancer trial, hopefully to commence in 2018. Quick word about what we're doing in terms of genetically engineering these cells. So let me shift now from an unmodified MIL with its inherent tumor specificity to a modified MIL, which we believe will retain its inherent specificity, but we will also add the recognition element of a CAR. We think there's a great opportunity here because several of the reasons for relapse in CAR T cells are potentially addressed by using this cell source. We've shown that MIL CARs can kill at lower levels of antigen than PBL CARs. I talked about the data that they persist. We're also generating data that shows that in addition to simply persisting, they also persist in function, whereas PBLs tend to diminish in function over time. And we're working to generate data that shows that if there is a complete loss of the cognate CAR antigen, that we may still have a safety net of inherent tumor specificity that could help diminish the frequency of relapse from antigen escape variants. And so this is the sort of data we're looking to show. I'm not going to dwell on this, but we're looking at systems both to show that the MIL CAR kills better than the PBL CAR in a cognate antigen positive system, as well as in models of antigen loss. So final 2 slides, just a few words about who is behind the company. Domain Associates, a well-known venture capital firm, Silver Rock and Fox Kaiser contributed $11 million roughly a year ago to get the company off the ground. Kim Noonan joined from Hopkins as a founder and our chief scientific officer. Steve Fishkoff, chief medical officer. Janice Phillips, we hired as one of our first hires to head up manufacturing, because that's such a critical piece of what everyone in this space needs to worry about. We have a very experienced board. Don Hayden was just named our chairman. He was the former president of Worldwide Pharmaceuticals at Bristol Myers. And then we've constructed the SAB to have a variety of perspectives. Ken Anderson is obviously the president of ASH and a myeloma expert. Yvonne as a founder. Marcella Maus is a cell therapy expert. Drew Pardall is a tumor immunology expert, and Jeff Weber is an immunotherapy for solid tumors expert. So we're trying to cover the basis of perspectives from each of the different components of what our technology does. And then I'll just close with what we're planning to do over the next 3 to 4 years. We think there are a lot of exciting milestones ahead of us. We should have a potential interim look. We're deciding exactly how deep that look is in mid-18 on the myeloma trial, and final data at the beginning of 2020. The lung trial we're designing should be, it's a Simon 2-stage design, which should report out data very quickly. We are budgeting for a second solid tumor trial. We're trying to pick the tumor type there based on preclinical data. And then we continue to work on the MIL cars, what we call MILS 4.0, which is to take our manufacturing process to the next level in terms of driving efficacy and specificity. We're right now consider ourselves on version 3 of the process. And then we're going to be doing a tech transfer in the next 6 months to a contract manufacturer. So with that, I'd like to thank you and appreciate your time this afternoon.